Hey people, let's talk about personalized medicine. Now, you might have noticed everybody looks a little bit different from everybody else. And this isn't just limited to your appearance or your personality, it also includes things like disease resistance and also how you respond to medication. So, up until now, you've probably talked a lot about genetics, which is the study of individual genes. But in recent years, we've moved on to genomics, which is studying all your genes together and how they work with environmental factors. So, over the years, the cost of genetic sequencing has gone way down. The first time we did this was the Human Genome Project from 1999 to 2003. That took a really long time, and it cost like $3 million. And now you can get your genome sequenced in a couple of weeks for like a couple $100. It's only going to get cheaper and faster as we go. So this now becomes a real possibility for general medical use. So 2007 is when we started doing these genome-wide analyses, where we're now looking for what genes and what even just DNA changes do we associate with certain diseases. And these are diseases that don't have a single gene cause, like cystic fibrosis. These are things like height or heart disease or breast cancer or Alzheimer's, where there's not a single gene, there's multiple things that affect these diseases, or just traits like height. So the goal here is personalized medicine, which is called pharmacogenomics. We're looking for individually tailored diagnoses, preventions, and treatments targeted at your specific genetic code. So here's a nice example of what it looks like to treat a population with a drug. And I want you to think about your most recent or most memorable legal drug that you have taken or that you know someone else has taken. Maybe this is Advil, maybe this is caffeine. And you might notice that for a lot of people in the population, you're with the green smiley faces. The drug is safe and it works. But for some people, you might be with the blue group, where the drug is safe, but it doesn't seem to do anything. If you're less lucky, you're going to be in the red group. This is where the drug is unsafe. It's giving you adverse side effects. And maybe it's also not doing its job. That's the worst group to be in. And maybe you're in the orange group where the drug is giving you bad side effects, but at least it's working. So think about the last time you took a legal drug. Which group were you in? And we think the differences here are because of genetics. So human drug response is something that actually affects a lot of people and costs a lot of money. In the US, which is the only place I could find stats for, about 1.5 million people a year are hospitalized for adverse drug interactions, so side effects. And it kills a lot of people. It also costs a whackload of money for the healthcare system. Some really common drugs, like antidepressant drugs, in somewhere between a quarter and half of all people who take them, we end up with really bad side effects. And so wouldn't it be great if we could figure out which people those were and give them a different drug? So here's some real life examples where this has happened. There is one gene in the body called CYP2D6, and it makes an enzyme that converts codeine, which is a narcotic drug, into morphine, a much stronger narcotic drug in your body. Now, most people do this at a certain rate, which means if we give you codeine, it gives you pain relief slowly. But there's some people that have a variant of this gene that makes them ultra fast at converting codeine to morphine. What that means is by giving them a normal dose of codeine, you could actually overdose them on morphine. And codeine and morphine are fat soluble, which means they actually can pass through breast milk into a baby. So in 2007, Health Canada issued a warning after a 13-day-old boy in Toronto died of a morphine overdose. The mom was almost arrested for killing her child until the doctors found that she was one of these ultra-fast metabolizers. So the doctors had given her a very normal dose of codeine after the birth, and it actually ended up killing her baby. In 2012, a 4-year-old First Nations boy in Northern Ontario also died of this same issue after being prescribed codeine from having his tonsils removed. So this is very serious. If you look here, we can see a breakdown based on different racial information of how much percentage of each population are these ultra rapid metabolizers, seen in kind of the maroon color. And you'll notice there's also a gray of how many are poor metabolizers. For these people, giving them a normal dose of codeine is not actually going to give them pain relief. Problem the other way too. Here's another example, Cisplatin. This is one of the very first cancer chemotherapy drugs. It was approved in 1978, and it really worked. It was amazing, except that in about 25% of adults and 60% of children, it resulted in permanent hearing loss. We later found out this is based on genetic factors. And nowadays, if you are being considered to treat your cancer with cisplatin, they will give you a genetic test to make sure that you're not one of the people who will end up suffering from hearing loss. Another one is Warfarin. This is a blood clotting drug. It basically prevents your blood from clotting, so it's a blood thinner. This is one of the most commonly prescribed drugs in both the US and Canada, but it's hard to find the right dose. If you give people too much of this blood thinner, their blood is so thin it doesn't clot and they can bleed out from a simple cut. If you give them too low a dose, they're going to form blood clots and they might die of a stroke or a heart attack. And it turns out the variation is responding from pretty much one gene. So most of the issues are the CYP2C9 gene. And in 2005, the FDA suggested that doctors should spend money testing people for this gene because it actually is cheaper to test people in advance for this gene than to spend money paying for their hospital stays when we get the dosage wrong. And there's currently lots and lots of clinical trials now where we are doing this genomic testing to see how different people respond to drugs based on their genetic profile. So can you do this? The answer is yes, depending. If there is a likely risk of something, like for example, breast cancer runs in your family, you can actually get tested for these gene variants right now. Or if you're going on a drug like cisplatin, for example, you can get tested right now. There's also some pilot studies currently happening at Sick Kids and Thornhill Medical Center about antidepressants and other kinds of drugs where we know certain genetic variances affect them a lot. If you want to just get tested because you can, you actually can pay to do that in Canada at Life Labs or Dynacare. And here's a list of a whole bunch of things you could get tested for if you want to pay for them. So what are we actually testing for here? Oh, you can also pay to get personal testing done. These are not medical grade. They don't count as medical information, although people treat them that way. But that's certainly something you can do as well. So what are we testing for? We're testing for SNPs, SNPs, single nucleotide polymorphisms. We're looking for parts of your DNA where there is a one-letter change between individuals. Remember that between me and any of you, we are 99 to 99.9% identical, no matter who you are. That's just human beings. So at most, there is a 1% difference between us. And at least, there's a 0.1% difference between us. And those differences are SNPs. Now, this may look like a mutation to you. We have arbitrarily set the threshold for something to be considered normal at 1% of the population. So you can see here the example, an A changed to a T. If 1% of the population or more has that, it's a SNP. If less than 1% of people have it, it's considered a mutation, which is maybe not a good thing. We've currently estimated there are about 10 million SNPs in the human genome, and that accounts for that 0.1% difference between people. Now, there are problems with this 1% rule. There are certain genetic disorders that seem to appear nearly at the 1% threshold or even above. Sickle cell anemia, for example, people with that allele, that's more than 1% of the human population. But people with the disease, so having two alleles, because it's a recessive disorder, that's less than 1% of the population. So is sickle cell a mutation or a SNP? It's hard to say. We've also found at least 40 SNPs that associate with type 2 diabetes. We don't know if they're a cause or an effect, we don't know, but we know that people who tend to get type 2 diabetes tend to have these 40 SNPs. So are those mutations, or should we still keep calling them SNPs? It's a question. So if we look at all the SNPs in your genotype, all the SNPs in your DNA, if we put all of those together, we call that your haplotype. And so this is the thing that we want to look at for people. Most of our DNA is identical, so it doesn't matter from this drug perspective. But the parts of us that are different, these SNPs, if I put them all together, that makes my haplotype. And one day in the future, your haplotype may be on your medical chart or on a chip on your health card so that your doctor can consult it when they're considering giving you a diagnosis or a treatment.